Okay, just a few words before we start seriously. Thank everybody for organizing this meeting. I think this is about the fourth or fifth time I've spoken in Ricken, and it's normally been an interesting discussion. I will go relatively slowly, I'm afraid, as this is being filmed and will be used for people maybe in institutes where there's not such an easy command of English. So please excuse me if you feel that I'm speaking too slowly. With all that said, I'd like to start on this introduction to the HFSP research programs. I will go slightly quickly through the first slides as Hideki has already mentioned some of the basic principles about the organization. And my aim, which is not so important here in Ricken, is to encourage people to get involved in international collaborations. But that's something which is a feature of Ricken, I know from my experience with scientists from your institutes. So, HFSP has three programs: fellowships, which are to fund young postdocs to go abroad. Then we have a small program, the Career Development Awards, which is to help those postdocs to come home and start setting up their first independent lab. And then the major part of HFSP support, which is what I'm responsible for, is the team grants, which are designed to initiate international collaborations. And I say very clearly initiate because we're looking for new ideas, new people coming together to look at a new way of looking at a problem in basic biological science. They're unfortunately not renewable, so there's a 3-year basic startup grant to get an interesting idea off the ground. And if that works, we have found that mostly national agencies then will kick in and start supporting those bits that they are concerned with. Okay, so we emphasize genuine international and preferably intercontinental collaborations. We're looking for innovative and interdisciplinary approaches, and 70% of HFSP funds go to scientists who are under 40, either through fellowships or the Young Investigator Program or the young team members in the senior grants. And most senior grants these days have at least one, if not two, young scientists as part of the team. The most important thing to avoid being disappointed is to understand the philosophy of the program. This is getting a new team together and putting forward a risky project, but one which you can argue might well work. And the job of HFSP, the mandate it was given right at the beginning, is try to encourage these very original projects. So we only finance basic research, which isn't a problem here. And it's essentially looking at the complex mechanisms of living organisms, which goes all the way from the subcellular, submolecular scale up to these days to highly complex problems in systems neurobiology, looking at the interaction of large numbers of individuals in a group showing group behavior. There are no priority areas. It's a bottom-up approach where scientists get together and say, I've got this great idea. I think the 2 or 3 of us could have a go at solving it. Let's try and get support from HFSP. As I say, we don't fund applied research. And so the particular problem is normally that people come in with clinical-based research, either to improve diagnostics or provide a treatment. That's slightly a problem because of the misnomer in the human frontier. People think it's medically orientated. In fact, the idea was, as you well know here, what are the limits of the human brain in working out things? That was the original concept. So the member countries, there are now 14 plus the European Union, which represents all of the smaller European countries. When setting up a grant, it's important that the team leader can only come from one of those member countries. The annual budget you heard was 54 million. The average grant value is just over $1 million. And our budget enables us to start 32 new grants each year on average. It's a tiny secretariat. There are only 16 people in the office, handling both the scientific aspects, the administrative aspects, and the public relations aspects. It's a very dedicated staff. All the people do their job very well and very rapidly. And I think if you ever have questions, don't hesitate to write or call. There are very friendly people who will get back to you with constructive answers within hours for an email, for example. It's very important that the project has a very clear aim and preferably a preferred methodology. Sometimes if some problems may come up and you may think, well, there's a couple of ways we could look at this. There's nothing wrong with proposing two ways of looking at it, but you must argue through why one of them is most likely to work. The funding is very simple. There are no budgets, which gets some administrators very annoyed. We make block grants so that a team of two will receive $250,000 a year for each of 3 years. 3 is $350,000, 4 it's $450,000, and we reduce those amounts if there are two team members from the same country. So, for example, if there are 3 and it's 2 in the same country, that drops to $300,000. The thing is to encourage international collaboration. The other thing is that the HFSP committees believe that small teams are the best teams. They prefer seeing 2 or 3 people working together rather than what we were seeing in the early days of HFSP, 7 or 8, where very clearly some of those team members were not going to be involved at all. What we're looking for is a real day-to-day collaboration between the team members, exchanging reagents, exchanging ideas, sharing results, discussing them, and then saying, what do we do next? I'm sorry, but as we're in a neuroscience institute, for some reason, neuroscientists claim, first of all, that they are the most broadly trained scientists possible, and yet they always need to put 5 or 6 of them into a team. This is something we see year after year after year. Those, unfortunately, nearly always are eliminated very early in the competition. We're very keen on neuroscience, but we like small teams in neuroscience just as well as in the other disciplines. So, all team members must be group leaders. We make an exception in Japan, as we know that becoming a professor in Japan may not occur until somebody's around 50. And as we want to encourage young scientists, in Japan we are happy if somebody has a letter from the head of their institute or whatever saying this young scientist is leading an independent small group. However small, it may be a postdoc and a student and a technician and the person themselves. We're quite happy to accept that as somebody being a group leader. We have to be fair to all countries and respect and respond to their different structures. For young investigators, all the team members must be within 5 years of being independent and no more than 10 years from their doctorate. There are allowances for parental leave, sickness, etc. All of that will be discussed very quickly and friendly with the office if you have any doubts. Then the program grants are for everyone else at any career stage, as long as they have the position of running a group. In fact, there are a lot of young scientists in the successful program grants. I think to date the youngest team leader of a program grant was 31, which is obviously very young. It was somebody who was very ambitious and who constructed a very convincing grant. We are encouraging particularly intercontinental collaborations. When HFSP was set up, the only Asian member was Japan, one of the most important founder members. But since then, we have added India, New Zealand, Korea, Australia, Singapore. So the whole group of now of Asian Pacific countries who are members, and we would like to see the position, their percentages increase over the next few years. I think it's inevitable. Seen from our small window of what's happening in world science, I would say the best science is moving towards Asia. It's leaving the States, it's crossing Europe and moving east. That's just where the very best modern innovative science is being done. The scientists themselves that we find amongst our awardees, we don't look at this at all during the review procedure when we actually have the list of, say, the 100 awardees in the grants. That's to say 30-odd grants, 3 people per team. That makes roughly 100 scientists each year. 30 to 40% of those are not working in their original country. There's something about the spirit of adventure as a scientist makes people move to where they can do their science, where the machine is, where the right group of people working in that area is. And that doesn't stop them moving back home later, but they are mobile. And actually, some of our awardees, quite a large number of our awardees, in the course of the award are headhunted by another institute in another country and move on again. And that's something we can handle. We can easily arrange for the grant to be transferred to another country, another institute. So, why is it important? I think it's probably obvious, but it's something I had to learn the hard way. Each country has a different way of training its youngsters in education, and then its students, and then its doctoral students. And this gives people different ways of thinking about problems. When I left Cambridge, where I've been working in English and speaking English and working in genetics, I moved to France, where I had to learn French and learn molecular biology. Those were 2 challenges. I was about 30 at the time. And I found actually it was a lot easier than I thought. It was actually very exciting. And I went on from there to work with people in something like 30 different nationalities. And I began to appreciate that each of those had a different way of looking at problems. Much later, I started to get involved in interdisciplinary research myself, working with computational biologists and with structural biologists. And there I realized there was another new set of languages. And one of the problems when you start one of these things is that initially people have got so used to their discipline. They do what I'm doing here. I'm talking at you because there's no feedback. And what you have to learn is in these interdisciplinary projects to teach particularly the young scientists to talk to each other. And in our case, it took 3 meetings of 3 groups to stimulate the youngsters to understand they couldn't just give their standard structural biology talk. They had to explain to the other team members what their limits were and what they were hoping to achieve. And after that, and that's something we see now in the final reports, scientists, including the team leaders, say that they've really learnt a lot more from this collaborative research. And for example, a biophysicist has given them a completely different way of looking at a problem. So, that's individual benefits. What about the career benefits? Our awards are dreadfully competitive, with something like a 3 to 4% success rate. And despite this, some of the very best scientists worldwide compete every year. They find it a challenge to get recognized year after year that they are amongst the leading group of scientists in the world. For younger scientists, the importance is that they've been assessed by an international panel and have been given a kind of a badge of quality, and that creates things for their career. Suddenly they find it much easier to get national funding, local funding. In our case, in my old laboratory in Strasbourg, suddenly the region were quite happy to contribute to the cost of a microscope or give a fellowship for a young student within this person's group. So it has a kind of cumulative effect. The review process is long, necessarily because it's very careful. Each year, at the end of March, first week in April, we receive something like 900 applications. These are seen remotely by the review committee members, of which there are 24 drawn from all of the participating countries. And they reduce the initial group of 900 down to about 300, which are discussed in Strasbourg, across the table each for 2.5 days in the summer. And those 300 lead to 80 or 90 of them being invited to submit a full application. The initial letter is only about 2 pages of science plus CVs, whereas a full application is something more like a regular application, maybe 15 pages of science plus the CVs and things. And that's what's going on at the moment in Strasbourg. We received 80, I think it was this year, and those have been sent out to male reviewers, experts in the different aspects of the project, so that by the end of the year, preparing for our January meeting, we should have 4 or 6 really detailed reviews regarding the quality, the riskiness of these applications. And there are surprising comments that go against a project. Very often people say this is excellent science, but it's the next logical step in what they've been doing for the last 10 or 15 years. And at that point, the committee will say, great science, but it's not for HFSP. There's no risk there. It's just what is the next thing to do. It'll probably work, may even have been done, which is obviously something we don't want to fund. We don't fund, like other agencies, experiments that are already dry in the notebooks at the time of the submission of the application. That's a fairly common practice in NIH, for example, is that you do the experiments, then apply for the money, then publish the experiments, do the next experiments, then apply for the next money, and so on. It's always a delayed process. This isn't a delayed process. This is things that haven't yet been started. This is just an idea that has come together and looks exciting. So, the idea is to bring in people from outside the life sciences, particularly computer sciences, chemists, physicists, biophysicists are very enthusiastic. They're probably the most imaginative group of people we see in the projects. The idea is to get some new ideas, risky ideas. There's no preliminary results required, and this must be science that is not going on anyway in the group. So what are the good ideas? Creating a project where there's a mix of approaches, a mix of expertise to the problem you have in mind. Keep to making a small team. That's to say 2 or 3 for a young investigator, 3 or 4 for a program grant. If you're invited to join a project, which often happens, only join when it's very clear how it's going to work. There is nothing worse than being an add-on to a project which is really something that's already being done and between existing collaborations. And there must be, if it's theory and experimental, there must be very clear forward feedback between the two approaches. Obviously, the bad ideas is setting up a project where everyone's doing roughly the same thing anyway. Setting up a project with too many research groups. Joining a group who have already collaborated extensively, and it's not clear what they want. Very often what's happened in the past is you will have two Americans who've been working together, and they'll say, oh, let's get someone from another country to make it international. And there's also been an unpleasant tendency to ask a Japanese colleague, because it's felt that Japan has had such an important influence in this program, there's an obvious choice. But very often the Japanese partner in the years gone by had no idea really what was expected and wasn't going to benefit from the collaboration, but was too polite to say no. And those projects from the last 15 years have been thrown out almost automatically when it's very clear that the third team member is an add-on to an existing collaboration. So avoid that like the plague. Similarly, there was a similar trick where the same kind of thing was that they were just trying to use resources that had been created by somebody, a CDA library, an antibody collection, a series of Drosophila mutants and so on. And there was no obvious feedback from to the person providing those reagents. The other aspect of that is a token add-on. Every year we have a couple of teams that have a Nobel Prize winner in it. But unfortunately, that is there for show mostly. It's very rare that that person is actually going to be making a new, novel contribution. We had it once. We had an 89-year-old Nobelist who led a team in the program grants, and whose project was judged the most impressive of the program grants that year, and they were actually placed #1 in the list. He was absolutely delighted that his ideas were still considered innovative, and we made a very happy Nobelist there. He really was over the moon about that. He had lots of prizes, lots of funding, but the HFSP badge for him, even then, meant a lot. It's rather more difficult if you're working in an area where you've been working perhaps with someone in Harvard, you've come back to Japan, and they've stayed in the US. You're thinking, well, this is international now. There's one of us on each side of the globe, Pacific. That is unlikely to be let through because it will be seen as an existing collaboration. There's not a new initiative there. Unless you start something completely different from what you had previously collaborated on, perhaps as postdocs in a lab in Harvard. That's the same, another aspect of the same problem. Sometimes it's possible, people have been in a large center. I've been saying Harvard, it could also be Cambridge, UK, where for some reason socially they have met as a plant biologist, a mathematician, and the move has happened. That is rather a more novel combination. We have one case of a plant biologist in Cambridge working with somebody in aerospatial research in California, and the idea was to look at the way that mutations in the plant seed changed the aerodynamics of the plant and altered its dispersal range. So in that case, that was considered fine because it was two people who had never worked together and had completely different expertise looking at something that neither of them would look at on their own. So HFSP has its niche of intercontinental collaboration. We target a very specialized group of applicants, people who are very keen on interdisciplinary research and have this passion for innovation. There's some people who questions come along like buses. There's always one coming along in the next hour or two if the one you've just thought of doesn't work. The biggest challenge is finding new partners, and the funding is small in some respects, but it's very competitive, and once you have one, it opens up possibilities in your career. One of the biggest errors is people just sending in what they're doing anyway, is what they've maybe just sent to the national agency, and they're just trying to put a few, what we would call whistles and bells on. They just add a few antibodies and have somebody look at the data. But when you get down to it and you read it, it's really what they should be doing themselves, and they're probably capable of making the antibodies and doing the analysis themselves. And just adding 2 other names to give it some kind of breadth and international flavor is quickly dismissed by the committee. There's a lot of content in the guidelines, and that explains what we're looking to fund. The grant office is very quick to respond to any inquiry, either on technical businesses about delays for parental leave, or whatever technical questions may come to mind, whether somebody is eligible for the young investigator. It's probably the most common request. And also you can send in 2 or 3 lines saying, I'm thinking of setting up a project on this topic with a biochemist, a computational biologist, and a structural biologist. What do you think? And there I will reply, if I can, saying, looks like a good idea, or I will say, it looks like 2 things we funded last year, so I don't think it'll be considered innovative anymore. And I'll give another couple of sentences to try and guide the thinking so that people don't spend time making an application that doesn't have a hope of being funded. There's a FAQ, frequently asked questions, which is updated every year, which responds to a lot of the general questions on how the process works. And you can see all of the abstracts of the funded applications for about the last 10 years, but it's really only worth looking at the last couple of years on our website. You go to the awards, you see a list of the awards with the team members, and underneath you can click on the abstract and see the abstract of each of those. So you might find 2 or 3 in your general area and find the kind of things we've been funding in that area. As I say, we're unlikely to fund 2 years running the same kind of thing. An example of what has been happening was 2 years ago we funded super microscope approaches to look at what was going on in cell culture, in flattened cell culture, basically, at membranes. We funded 2 or 3 of those in 1 year because we weren't sure whose approach was going to be the most effective. This year, or last year, we haven't looked at those. We've been looking at things where the microscopy must go deeper down into an organoid or into a living animal. We're still very interested in the development of those microscope techniques, but we've moved on from just a flattened cell on a slide into something that has more of a life form than that. We sometimes get very generic projects. We have what I've called I've Got a Mouse, which was a year when I think we had 50 different applications, which could be resumed as I'm making a transgenic mouse. I have a friend who does biochemistry. I have a friend who does anatomy. We'll look at the consequences. And that was the same 50 times. And of course, if I can make that short abstract of what the project is about, it means it's not an original project. I think last year I have an organoid, a cell line, or a mutant animal. I'm going to look at the biochemistry of this pathway. I'm going to make an antibody against the critical member of the pathway, an enzyme or whatever, and I'm going to cure something. Which was fine, but that's something that had already got into the clinic. People were already using antibody treatments for cancer. So although each of those projects was very valuable for national funding agencies, very critical for medical agencies in particular, it wasn't an HFSP level project because it was no longer an original idea. It was do A, do B, do C, do D. Okay. Now, how do you write a good letter of intent? It has to be simple and clear. This is what I want to do. This is the combination of expertise which will enable me to do this, and this is what we hope to find along the way. For HFSP, it's hopeless to try and recycle something from another grant application. The language should be very simple. Reviewers get annoyed with people trying to kind of disguise what's in their project by adding rather sophisticated sentences which don't actually mean anything. There's a terrible tendency these days. And most importantly, once you've written it, have someone in the field, or in not exactly in the field, but alongside, read it and see if they understand it. If they don't understand instinctively your abbreviations or something you say, then clearly a reviewer who's going to be even further away won't understand. I say that in a way I'm pleading because I read 900 of these things each year, and there's nothing more aggravating because it's so badly written that you can't understand it. You read something, great idea, great approach, I haven't seen that before, and you get the feeling that's going to be in the last 10%, which is going into the full applications. And I can tell you it's a relief to read those when you've read 9 that are really rather messy. This kind of thing gets not only me, but the whole of the committee annoyed. There are some people who are applying to a a program that asks for interdisciplinary international collaboration and innovation, and they feel they have to write that, and not only write it, but put it in bold letters 3 or 4 times in this 2 pages. That looks very silly and very immature. The committee gets very annoyed because they say, no, all of them should be that. You don't need to tell me it. That's the kind of thing I was saying you shouldn't add unnecessarily to your argument. The boxes for writing an application online are of a fixed length, but you don't have to fill them. If you've said very clearly what you intend to do in a section, just stop there. Don't start trying to write other things in there. That's again aggravating for reviewers. On the other hand, if you get through to full application, you should give your intended strategy, what you think is going to work. You might discuss where there might be little problems, where there might be a bottleneck or a problem, and in that case you must write how you think you can get round. What's an alternative approach to get round this particular problem? I don't think anyone here will do this kind of nonsense, but there are some senior scientists who feel that instead of writing in what they're actually going to do, they'll spend all of the space repeating what is actually in their CV, which we have behind the scientific project. What we need to know is what will they do? How will they contribute to the collaboration? We're not interested in how many students they've trained in the past, nor even really how many publications they have. They have that in their CV. The other error is to just give a catalogue of approaches that might be useful. And a third one that is probably a little bit better now. This was a problem about 3 or 4 years ago when we started getting a lot of systems biology. People were going to analyze X genes in Y species, Z habitats. It was a dimensional problem, and it was possible that with the advances in omics that they would get all this data, but they gave no suggestion as to how they would be able to treat that data. And so a lot of them went out of the window just because we're not there just to pay for omics experiments. Young investigators tend to try and disguise their own work as a team effort. This is again, in the full application, we often see a listing of techniques, and those are things that reviewers have written, saying this is a shopping list, a catalogue, they're throwing the toolbox at it, it's a laundry list, it's a lifetime project. We don't mind, these are 3-year grants, we don't mind people putting in a project that will actually take 5 years. That's reasonable. But when anyone who knows anything about it realizes it's a 10-year effort or a 15-year effort, they're just saying they haven't thought this through. This is unrealistic. And we had a bad period. I think it's a little better now, is that male reviewers were finding that young scientists didn't seem to know what had been going on in their field 5 years ago. As someone who was a postdoc had to walk from library to library to do their biblio, and today you can do it on the web just by a few clicks, it seems incredible that people are not aware sometimes that this experiment has been done 3 or 4 years ago, and the result is this, and this project, this is something I've seen 3 or 4 times, this project will not actually alter that conclusion. They're using more sophisticated technologies, but the answer is already well known in the literature, as senior scientists tend to say. So, male reviewers, we try and get the best. We get a very good response from male reviewers. They very often say they're honored to have been invited to review an HFSP project. I've even had someone who wrote back and said, I've been on NIH panels for over, I think it was 20 years, and this is the most exciting project I've ever had to review, which is obviously very gratifying for the quality of work that we are receiving at HFSP. We're not an agency that dismisses something if there's one negative review. The committee meeting in January is very lively, and the 2 committee members who are piloting each application, armed with the external reviews, will discuss very intensively. And in fact, our members of those committees are very broad scientists, and 5 or 6 of the committee, let's say a quarter, will talk about any one application. There are 2 people who are piloting it, but there'll always be 2 or 3 others, maybe more, who have something to say. They know one of the teams, and they're convinced or not convinced by their approach, or someone has been and heard the work at a conference recently, and they feel that this is already well established, this is not a new project. The last one is really about being sure about the people you choose. We invite full applications, the teams to suggest 2 or 3 people who are appropriate in their field as reviewers. I check them to make sure they're scientifically sound before adding them to the list. They make up maybe 1 or 2 out of the 6 reviews that are requested. I also request reviews from people who are not cited by the applicants, and the review committee members piloting also suggest people that they feel are appropriate in different fields of the application. And unfortunately, 1 or 2 times each year, there are teams that are destroyed by people they have suggested, who they thought respected their work, but found it really rather light, and it's not critical remarks as such. It's in the sense of being unpleasant. It's just a very detailed analysis of the application, pointing out its weaknesses. And obviously, if you cite some of the best people in the field, they're most likely to have a very keen, critical eye on any experiment. That's just a behavior graph. That's scientists behaving 3 years running. It's the way they respond to deadlines. I started this as a joke about 10 years ago, and it turns out to be true for all programs. The only thing is that the fellowship curves are even sharper. This is the last few days. We get 2/3 of the applications in the last 48 hours. The fellows, probably it's over a half in the last 24 hours. So people learn a little bit with time to not really have to do it on the deadline, but it doesn't get that much better. We just live with it. Actually, we use the curves to make sure that things are going correctly. If there's a hick somewhere, if this isn't going up on one of those curves, we're worried there's a problem with the machine, or there's an outside influence, which happened a couple of years ago, where a company had started sending mass mailings to scientists the world over, inviting them to apply to HFSP under completely false pretenses. Luckily, because of these curves, I spotted the error, and within 24 hours wrote to the company and told them to stop it. And fortunately that happened, but they still managed to get an extra 300 applications sent to us because of their mass mailings. Okay, so contact information for the grants is there. You want me to go on a little bit? Okay. So I've been asked also to just give a general outline on fellowships, because obviously being in a Japanese laboratory, you can both send your students anywhere in the world and receive students from anywhere in the world on our long-term fellowships. There are two types. There's the traditional long-term fellowship, where people have done a thesis in biology and no more than 3 years previously, and proposed to go to a different country and work in a different field so as to expand their scientific training. The other group are the cross-disciplinary fellowships, where they have typically degrees in mathematics, physics, chemistry, robotics, and are interested in taking part in a biology project. Now, we feel that they are a very stimulating source of new ideas to projects, and it's an area that we would like to develop further. At the moment, it represents only 10% of our fellowships. We're much happier to see that around 20-25%. Maybe it's an opportunity to bring somebody trained in robotics into a neurobiology and neuroscience project. So, there are normally 3-year fellowships. There's a living allowance, travel and child and parental leave allowances. The 3rd year is often used for repatriation to the home country, and the final year, the 3rd year, can be kept for up to 2 years. So what happens, particularly in the States, is people go, use 2 years of their fellowship. The host then pays them, because they're so interested in this fellow, for 2 years off their lab funds, and then they have the 3rd year of fellowship to use to go back to their home country or to move on to somewhere else. And the fellows find this particularly exciting. It's a very open fellowship system. The other advantage is that HFSP insists on a level of independence in the project that the fellow proposes. It mustn't be simply an obvious ongoing piece of work in the host lab. It must be something where it's clear you can see what the HFSP fellow is going to bring to the project, bring to the lab's work. It has a slightly better success rate than the grants. We get roughly 800 applications and make 80 awards, so it's roughly a 10% success rate. So, I must admit these slides were lent to me by a colleague, and I'm discovering them to a certain extent. Obviously, it's to provide international research opportunities, to broaden expertise. As I said, even for the biologists staying in biology, they must switch fields. They should learn new theories. They should be working in a lab which publishes in other journals from the ones that they have been brought up with. It must be basic questions. And the idea is to give them the support to become an independent scientist. I think it's very difficult to be independent in your home lab, so to speak, where you've done your studies and then have become a PhD. It's going abroad, it's going to do this kind of research that will strengthen the character of scientists to become independent scientists. And the advice is that if it's something that's going to work, it has to be basic research, creative, frontier, potential to be transformative. I suppose, yes, it's for the whole field. I'm not quite up to some of this modern language. I'm a bit worried by words like transformative. It's got to be great science, basically. Japan has always been a fairly strong candidate in these fellowships, behind France, Germany. India has a lot of applicants, but not so many awards. And Spain went through a period. Japan, we'd like to see more activity. We know it's a general problem that Japanese scientists have become reluctant to leave and go abroad. We used to see many more applications from Japan 5 or 10 years ago. I know it's a matter of economic stress at home. They're worried if you leave, you'll never come back. That was the worry of a lot of the French students in the lab I was in for Strasbourg for over 20 years, until actually the French government made it compulsory, an overseas stay, before you could get a permanent position in France. Everyone goes to the USA. Our problem is trying to get them back to the other countries, which begins to work. I think probably they'll not be so keen to go to the USA these next few years. Japan's here. We'd love to see more people coming to Japan. Try and encourage them into your laboratories. There's a lot of talent out there. That's the success rate, as I say, it's around 10% all the time. This program is a slightly different time of the year. It starts in August, and by late August, the applications have to be made, and the results are announced in March. Career Development Awards, very brief, very small program. The idea is to make the fellowship even more interesting because it's a limited competition where only former HFSP fellows within a certain career stage can apply. We normally have about 50 applications. This provides $300,000 over 3 years, so it's roughly $100,000 a year. It helps people set up their first independent laboratory. It can be taken by coming home or by moving from the fellowship to another member country. That was to make sure that all of the people who went to the States didn't stay in the States. They could make this application as long as they moved to another country. The important thing about this money is, like all HFSP money, it's supple, I guess the word is. You can use it for what you need. There are no, as I said, for the grants, there's no prefix budget. There's a list of things it can be used for. And just as the scientists in the grant teams can use their money where they think it will best advance the project, the youngsters setting up their labs with the CDAs have the same possibility. And soft money, that's the word. Soft money is so important to innovative scientific research. I know that because I spent over 30 years in labs pushing the frontiers back, and the importance there was to be able to do what you needed to do when you needed to do it. We were lucky to have good funding, and there was always this soft money available to buy a piece of apparatus, to hire somebody, to help a student who had 6 months more work to do. And unfortunately, most grant schemes really look at everything in detail and report everything in detail. That's not the HFSP spirit. The spirit is getting science done the best way possible and trusting the team leader to use the money to the best possible advantage. So, those two programs are both run by my colleague, Dr. Pauli, who's been with us now just over a year. And contact details for all of that is also in the handout. With that, I hope I have finished. Thank you very much for your attention.
Thank you very much for your presentation. It's very, very useful for us. And my question is the HFSP project. There are 3 main programs. The one is a fellowship, and the second is a career development award, and the third is a research grant. My question is about a research grant. There are 2 programs. One is program grants, and the second is young investors grants. And I would like to ask you the image of a young investigator. In this paper, there is a comment of young investigator is within 5 years of starting their first independent positions. What is the image of this independent position? Is it a PI or non-PI, but he has his project in, he belongs to another, his boss?
Yes, I think that's the most delicate question because we treat it on a country-by-country basis. So in the States, it's very easy. Somebody finishes a postdoc and gets offered an assistant professorship somewhere. They change lab, they get startup funds, and they apply to us saying, I'm now independent. We know very well that in Japan, it's very difficult for anyone to get that. But we wouldn't in any way want them excluded, which is why we ask if you feel that you have scientific independence, that you're not just running a project for your supervisor, then we're very happy to take the group leader's word for that he will write and say, Dr. So-and-so is technically in my group, but is running an independent project with the assistance of, and then just naming a small 1 or 2 people who work with him or her. I think that's very important. Similarly, in Germany, we have the same problem. We have people who apply from Max Planck institutes, and there are some Max Planck directors who will just put 4 heads of department, and then everyone else underneath is just a list, whereas others will make subdivisions within the department and say project leader or theme leader or whatever. And there we accept them as well as independent scientists because they're responsible scientifically for the project that's going on. That's the real thing. Is 2 things, is that the young scientist is not only intellectually responsible for the project, but also will have control of the funds. This was brought in because there were some of the big labs that were trying to use their junior members just as a means of pulling more money into the pot. And that was not what HFSP was set up to help the very big labs just increase their general funds. This is really to get behind individual investigators in their projects, their dreams, if you like. It's a very important question.
Okay, thank you very much. It's very useful to understand the philosophy of HFSP. They how to say in English, they they are very, very good for the young investigator and challenging project. Thank you very much.
My question is not very major question, but I'm interested in the program grants.
Mhm.
And you have mentioned about the case that sometimes in the review process, the proposal or the grant is synced by the reviewers. And unfortunately, the applicant will apply for the second round, naming the same candidate reviewers.
What I
And-
try and do
yeah.
there is not use those suggestions, because I don't think it's fair. I try and find other reviewers. I choose amongst the suggested reviewers.
Yeah.
And unfortunately, as you say, in the first application, maybe out of the 6 they suggested, there have been 2 that have been, or 1 even, that's been very critical. If they will tend to suggest that person the next year, but I won't use them. I will ask other people, because I don't think it's fair to go back
Yes.
either for the candidate or even for the person who's given their opinion.
Mhm.
That's just my idea of fairness.
Okay, okay. Because for the applicant side,
You don't know.
Oh,
No,
yeah.
exactly.
Thank you very much.
It's an important question, and I already have a way of treating it.
Anything else? Is there any question?
So, for the better understanding by the audience or video film watchers, I would like to ask you that you have explained that the HFSPO will welcome novel and innovative research applications. But are there any clear definition? What is human frontier science? So, are there any? Do we have any glossary for the exact meaning of the human frontier science?
I think the problem is that the frontiers are moving. If they're not, we do go through periods of stagnation. I can remember, for example, when I first joined HFSP, developmental biology was totally stagnant for about 4 years. It had reached a point where everybody was doing the same thing, and it required somebody starting working with biophysicists and computational biologists, looking at measuring molecules that were being used in early embryogenesis to cause that embryogenesis to function. And then there was a new takeoff. But it's very often the case that you get the feeling that, for example, we had optogenetics. Optogenetics in neurobiology came in, and then everybody was using it for a year or 2. Then you have to have someone else who gets another way of using it or another idea. That's when the frontiers move. And it's a little bit of a jerky process, but that's what science is about. And it's those pioneers who just kind of wake up in the night saying, hey, why don't we look at it this way, that suddenly move everybody on in that field. What's very clear is, let's look at the CRISPR story. The first year that was being worked on, we had 2 applications using CRISPR. The next year we had 50, and the third year we had 350, because at that stage, the CRISPR technology had become like PCR or a restriction enzyme many years ago, and it had just become a regular element of any modern science. And it's very interesting. That's one reason that I've stayed so long working with HFSP, is because the science is moving. You feel it moving every year. If it wasn't moving, I frankly would have got bored after 3 or 4 years. That would have been quite enough. But it's always exciting seeing these new ideas coming in. I'm afraid there's no real definition, because something that I would have gone wow to 2 years ago no longer excites me. I can remember one member of the important summer committee saying, oh, another magnetic tweezers project, for something that 2 years ago he would have been excited about. That's the way science works. Some people make breakthroughs. I was, well, no, it's a Greek thing. I don't know how many of you have been to Greece, but the Greeks build their houses and then they leave pillars going up. And when they have enough money, they then fill in with bricks. So I always think that HFSP should be funding the people who put the pillars up. We have enough good bricklayers in the world who fill in with the routine science. And then when we get to that point, we need someone else to put some more pillars up. And I think that's the way science happens. We need to be encouraging those people who have original ideas, and that's what HFSP tries to do. And it's getting more and more complicated, and it requires the input of different disciplines. Okay, thank you.